Hey everyone, good morning for everyone outside of India and good evening for people in India. Hope I can finish it in an hour. So, because I don't know whether you guys had dinner or not. I'm doing small talk because I know people are still joining. When I say 7:30, it is usually Indian Standard Time, right? So it is 7:35, 7:36. Doesn't matter, it's evening or morning. But I do see a lot of people have joined. People who are not part of MSc 09 batch, please sign in anyway because I think just listening to your hi ma'am, hi and all that goes a long way at a time when we are all isolated in our homes. So just say hi. You can even say hi everyone. That way everyone who joins gets your hi, okay? They can all see your hi on the chat screen. So it's pretty good way to stay connected and be informed. Okay. So I'll probably take few more minutes until the stream of people joining slows down a little bit. Maybe now it is 7:32 a.m. and Pacific Standard Time. I'll start in 3 minutes, 7:35. Okay, so we'll wait a little bit. I'm going to mute for just few more minutes. Okay, so I see that the stream of people who are joining has slowed down. According to my count here, there are 54 people who have joined this stream, which is good because from MSc 09 itself we have 40 people. So some seniors and some other people who are not at IBAB and doing some senior fellowship and everything, they also join. So in any event, the reason why I took the topic that I'm going to present today is because we all have many questions in our head. I know most of us are looking at Google to find answer regarding this COVID-19, and we find some answer, we don't find some answer. I found answers for many questions. I could not find answers for many questions that even I have in mind. So obviously I don't have answers for all the questions you may have in mind. So this session, this 1 hour session, I'm thinking we will come up with multiple questions. We can collect all the questions and work towards it together. So the reason why I'm going to call this, I'm going to switch now to my PowerPoint window and in a few minutes, and I will say that this lecture is specifically for people in bioinformatics because this is the bioinformatics class. I'm sure others who joined may also be in biological sciences, so they will get it. Some places you may not understand, and that is reasonable. So don't think that you're lagging behind. We are all lagging behind when things like this happen, right? So I'm calling this what bioinformatics can do at a time like this. So I'm calling this lecture. I'm going to switch to hold on one second. I'm going to switch to my PowerPoint presentation where you will see in a few seconds real world bioinformatics. At a time when the whole world is going toward data science. So, what is real world bioinformatics? Means, is there a real world application that bioinformatics, all the lessons you're taking in MSc 09 classes, like second semester, and you have not started third semester, I will probably start third semester lectures right away, because some of the lecturers may not be able to use the online lectures. So in second semester and third semester, you learn a lot in bioinformatics. So what I want to accomplish today is to see how what you're learning today and what you will be learning in the next 8 or 9 months, how it's going to apply to a pandemic that's happening in the world today, and not to overwhelm you with COVID, because I know everybody is full of news in the area of COVID. But this will tell you how to mitigate all those information and confusion in your head by learning more. Okay, so real world means what is happening in the real world today and what bioinformatics can do. So going to the next slide. In this slide, of course, 2019 nCoV, which is a virus, which is also called SARS-CoV-2, because we had SARS-CoV before. I will talk about that first to know what we learned from that epidemic back in 2002, 2003, and now come to see how we can use that to understand COVID-19, right? So you see that of course, COVID-19 looks beautiful, right? Thanks God it's microscopic. Otherwise, we would have this in our drawing room, in our flower vases by now, and getting sick, right? So it's microscopic, but it does look beautiful. Of course, the color is how we colored it. It is not as colorful when you look through the microscope. But in any event, we have a lot of questions. Of course, this is not, I'm not listing all the questions that come to your mind, but of course, the first question we all heard, oh, what is coronavirus? Of course, I have not heard about it before, and many of you didn't, but people who are microbiologists and virologists, they probably heard about it. So we suddenly got this new terminology we call COVID-19, SARS-CoV-19, all those names are coming up. But what is coronavirus? And the second question is coronavirus new? Of course, it is not new. So, but because we are hearing it for the first time, it is new for us, right? So the coronavirus do come. Now we know from the 2002 SARS epidemic that coronavirus has come from bat to animal first, and animals get sick, and then from animal it goes to human, and in the last 20 years this has happened 3 times, bat to animal to human. What do we know and what can we learn from this? Because it has happened in 2002, 18 years ago, and then it happened 8 years ago, and now it's happening. So how can we be even prepared? How come we were not prepared? Okay, can we even be prepared? I was getting mad thinking that why didn't we have drugs already for COVID-19? We will get an answer to these questions. And the next question I'm asking is, how do we know it came from bat? How did we find out? Because it's not like we live next to bat every day of our life. How did we even make that connection? And as I'm reading this question, I request you guys, if you have a question in your head, just type it in. You just, now that you know how to type it in with hi ma'am, hello ma'am, you just type your question. So by the end of the lecture, we can see whether we found answers to all of your lectures, all your questions. Otherwise, we can have another lecture to answering all your questions, or you can do research yourself. And the next question that I had in mind was, do the bats get sick with coronavirus? So we know that coronavirus is there in bats. I will tell you how we found out. But do they get sick? That's the next question. When the animals get coronavirus, do they get sick? Is it possible that SARS-CoV-2, which is the name of the virus that causes COVID-19, COVID-19 is the name of the disease, and SARS-CoV-2 we call is okay. So SARS-CoV-2 is a so next question is SARS-CoV-2 a lab strain? Okay, that is a question that everybody immediately asks, because it started in one place where there could be some labs people are working on. Did it escape from a lab? And how did it escape and become a pandemic so quickly, either from lab or from bat? How did it escape? And then, of course, all your questions are right here after that. These are my questions, and because of these questions, I did some research and I wanted to share that with you today. So going to the next slide. The first question we are asking is coronavirus new to infect human? The answer is no. Answer is no. So this SARS-CoV-2 or COVID-19 that causes COVID-19 is of course one of the viruses that is discovered in 2019, and actually December or early January of 2020 we know this virus, we have isolated it. And the second is SARS-CoV, which is also called SARS-CoV-1 sometimes. It caused SARS, which is in the second column, and that epidemic happened in 2003. Everyone in the lab, probably in the lecture right now, you guys were very young, probably you were just 2, 3 years, 4, 5 years old. At that time, you may not remember, but I'm old enough to remember and got very scared about this SARS thing that started again in a market in China. Okay, and then there was a MERS-CoV, which was a Middle Eastern. SARS stands for severe upper respiratory syndrome. Okay, so MERS, which is a Middle Eastern respiratory syndrome, that came in 2012, and that, it wasn't that big an epidemic, but it did affect a population. And then we now know that many of the pneumonia, many older people, when they get pneumonia, and some of them are getting it through other coronaviruses. Those strains are called HKU1, NL63, OC43, 229E. So the coronavirus may not be new, but the knowledge of coronavirus causing pneumonia and upper respiratory distress is new. That we learned only through SARS epidemic in 2003. Okay, so now we know there may be many coronaviruses. So I will tell you how we found out. So I'm going to go back now in the next slide to what do we know from the SARS epidemic in 2003, and then we can come back to understanding the COVID-19 pandemic, I mean. So in the next slide, so these are all the things that we know happened. Now we have gathered enough information because of the SARS infection, SARS epidemic in 2003. So what are all the things we know? We know that there is the NL63 on the top, on the right hand thing. We are saying how it came from bat to animal to human, or in some cases rat to animal to human. So we now know that NL63, which causes pneumonia, came from bat. We don't know which animal, but it went to human. Then there is a 229E strain of coronavirus that also came from bat to an animal to human. But you see this arrow, that's orange arrow, that tells you that there's a very mild symptoms. Of course, when it comes to older people, they get very sick, but these are very mild pneumonia causing viruses. So, and then you see in the below, I circled it in red because SARS-CoV is the first one that really helped us to find out the origin of coronaviruses, which will help us to understand COVID-19 and which will also make us be humble about can we protect us from future onslaught of COVID viruses coming and causing a pandemic. Okay, so in the case of SARS-CoV, right here, it came from bat and it went to an animal called civet. These are actually raccoon-like, something between maybe skunk or some kind of, these are all mammals, of course. They look somewhere between the rat family and dog family. So I have some pictures of that I can show you. Okay, but how we found out is the SARS-CoV was first isolated from a patient late 2002, early 2003. We sequenced SARS-CoV and now we are tracing it back. How did we get it? So first SARS-CoV infection was found in people who were in the market, a market in China. And so of course we asked them where they had been. So we know they were in that market. We also knew that they were handling civets. So in these markets there are a lot of live animals that are maintained, exotic live animals, and civets were one of them, and they were handling civets. So the next thing, obvious thing to look, do the civets, because now you know the SARS-CoV genome, because we just got it from a patient and we sequenced it. Because you know the genome, that's where bioinformatics comes in. Because you have a genome, you go into isolating, looking in civets whether they are infected, whether they have coronavirus. And so you can isolate those coronavirus genome and sequence and see whether they're similar from what we got from the patient. And then, of course, this is a wild jump that how did the civets get sick? So on the left, I have some questions from this kind of picture. When did we find out about this? How did we find out? That I'm curious. How do we make all these connections? These are crazy connections, correct? So how do we make these connections? How many years or who worked on these things? Who was involved? And if you want to search Google, you can search by a Chinese woman who is known as Batwoman, who hunted all the bats in the caves, and she was able to make this connection for the first time. So it really took, how do you go in the caves and find out coronavirus in the bat and sequence them and make this connection? This was very hard and tough work, and people, researchers are crazy, as you know already, right? So it took 5 years of hunt in remote caves, and they found 11 species according to the paper published yearly in this decade. There may be many more people have found, and bats are a hotbed for coronaviruses. From her research, we know that. Okay. And we also know that these bat viruses and recombine because they exist in bat without making bat sick. They keep recombining, and of course in bat also it is there in their lung and it's there in their mouth and it comes in the feces. Okay, so they can recombine to create novel viruses. This is the scary part. Okay. So in the next slide I'm going to show you Bats, bats, bats. Okay, now we're worried about bats. Why do bats carry these viruses? And why do we, I don't know how these bat women or some other researchers or virologists who work day and night about this microbiome and virome, how did they make this connection that from civet to bat? Because civet, you can see that it's an easy connection because people were handling that animal and you can see that. But how did they make this connection to bat? Okay. But one of the thing of bats which is special in the entire mammalian family is bats are the only mammals that fly, so they are exposed to many environmental conditions. Other thing that is different between bat and other mammals is flying costs a lot of energy. This is very interesting for me to know that their body temperature raised 2 times in a day, resembling fever. One while eating and one roosting. What is the relevance of that? Why I am talking about it? Because these coronaviruses that are maintained or symbiotically sitting in bats, they are used to high temperatures. They live in high temperatures. So these temperatures have made the symbiont viruses to handle high fever, mean that high fever in human being cannot kill it, which is resisting one of our first defense against infection. Why we get sick and we get very high fever in coronavirus infection. Bats do not get sick from coronaviruses. I still need to find out why. And it's believed to be a natural host or a hotbed for coronaviruses in future. Coronavirus directly from bat does not infect human. We know that. How do we know? Because you can do experiments. In this class and in my senior class who may be attending this lecture now, you all learn experiments to do in molecular biology experiment and in vitro, how we can take human cells in vitro and how you can drop these coronaviruses and see whether it infects the cells. You can do biochemically, you can measure it biochemically, you can do it experimentally. So they have done all this and they know that it doesn't infect human cells directly. But a spillover is a term used when viruses are being shed by animals and other animals get it. That's a spillover. So in nature, spillovers do happen, and bats can spill over to other animals through their feces, or when some animals may eat their feces and then they can get, or they can shed when they get stressed by human activities, which I have to say is in this market setting and in other wild forest, we are encroaching on their landscape and they get stressed, and when they're stressed they spill over the viruses they have. One of the examples people can ask, what stress? These days whenever we talk about stress, we are talking about mental stress, office stress, and somebody is stressing me out by saying all this. But that is not the only stress, right? Of course, in the animal world, that may not be the stress. They get stressed because we are encroaching in their property. And one example I can give you is when you go in cold climate. Of course, not many of us may be aware of it. When we go in cold climate, your nose starts dripping. Okay, so temperature stress, other stress can make your nose start dripping. So if you were unknowingly holding your coronavirus in your body, it will come out during stress. So that's one way to look at why the bats will be spilling over during stress. Okay, that's just a simplistic way of looking at it. There's more to learn in this process. So in the next slide, Okay, so the important thing is how do we trace back using bioinformatics, using genomics? How do we trace back something that came from bat and rat? Even if you make this connection, how do you know that came from that? Can we use our bioinformatics to find out? I'm just getting some messages saying I'm unable to join. You know, if you're unable to join, that's okay, because I think this video perhaps is getting recorded and I will make it available via YouTube. Okay, so don't worry about it. So SARS, which was the 2002-2003, I was too impressed with so much research that went on into finding what really happened. And of course, you cannot get prepared for again a pandemic or an epidemic, but we can quickly learn what could have happened. That's the best thing that happened. So let me go to see what happened when we got SARS in 2002. Okay, so people who got initially infected, they said they went to the market, they said they were handling civets. So of course the obvious source of infection was civet. What did they do? They hunt from market to remote caves in Yunnan to find out what could have happened. How did they find out? Viruses were identified in 91 civets in the market. Okay, 91 civets were there in the market at that time. They went and looking for coronavirus in those civets. How would you find out which civet has coronavirus? That's also fairly simple, right? Okay, I'm not going to ask you to guess because I cannot hear you back. If you were in a class right in front of me, I would have asked you to guess. So I want you to think a little bit. Think of how you would do it. What was the sequence of events why we went to civets? We had the human virus. So you know humans were infected. And so you have the antibody that from the human serum that binds to the virus. Okay, so you can make an antibody panel and you can screen the serums from these civets, right? To see whether the civets have the viruses. That's quick. And then select for the civet that's so positive for the antibody. Are you getting it? Okay, this is how you find out whether you have immunity or not, right? By screening for the presence of an antibody. So the same they did for civets. There were 91 civets they took, and they took also 15 raccoon dogs which were next to the civets, and some of the people who were sick, they were also handling raccoon dogs. I put the figures of these two animals so that you know we may not be familiar with that. They all look like some kind of a rodent family, okay? Rodent and dog, yeah? So, okay, so they found coronavirus in all 91 civets, and not only that, when they sequence the genome, of course, as a bioinformatician, you are immediately asking, do the genome of this civet coronavirus resemble the one that we sequenced from patients? The answer is yes, 100%. Okay. And then the raccoon dogs also had coronavirus, and their genomes also resembled. I'll show you in a bit the genome similarity between that and the SARS-CoV back in 2003-2004. Okay, and then the question is, okay, so are civets and the raccoon dog the natural carriers of this virus? How would we know? Unless you go to the wild and find out from these coronavirus there in the wild civet. So you have to be impressed with the amount of experiment that has been done. They have taken 1,107 civets, sampled from farms spread over 12 provinces in China, which were claimed to be source of the traded animals that were there in the market. Okay, so if the wild civets had the virus, then you know that civets are the original culprit and they are the one who are hotbed for coronavirus. But that's not what happened. None of the 1,107 civets had tested positive for coronavirus. using the antibody test, of course, right? Okay. Now, in 2005, okay, I don't know how the jump happened from, okay, of course, none of the other market animals had it. So, the jump could have happened because virologists may have a hunch because they work day in and day out with these things. So, I'm still trying to find out how that connection was made to bats. I urge you guys to do that research, and if you find an answer, let me know. But I think if you're in a field, when you've worked a long time in a particular field, and like I'm in bioinformatics, I've done computational biology all my life, I can make some connections. It's hard to explain. Okay, so I'm thinking maybe that's how they made that connection. So they thought maybe bats are the culprit, or otherwise they may also know because these animals may eat bat maybe in the wild. Okay, that may be another reason. So you need to know the animal behavior and other source of viruses and everything to make that connection. So there was this bat woman, they call it. She went into caves. She worked 5 years into caves, very, very narrow caves she will go, and she found out that the horseshoe bats from Yunnan province are the hotbed for novel coronaviruses. And they have many coronaviruses, at least 11 is known now, 11 species, and none of them are similar to SARS-CoV. You were expecting like all of them should be similar to SARS-CoV. None of them are similar to SARS-CoV. Until 2013, we did not find a coronavirus from bat that even bound to the same receptor that SARS-CoV bound in human. So we were not sure what's going on, but we know coronavirus is a hotbed. And later from lab experiment we know that recombination happens in coronavirus as inside bat. So maybe some recombination which is naturally happening gets selected to infect animals or humans, whoever come in contact with it. So recombination event, the recombination is a combinatorics, right? Any amount of recombination can be happening in nature. It's hard to predict. So one of the things you can get scared here is what is the next recombination that's going to make next epidemic or next pandemic in human or animals. That's a scary thing. But for now we know a recombination between two bat strain names. Don't worry about it. If you're interested in it, you can look into it. But two strains recombine to make a spike protein that's called new strain SZ3, which we believe is the start of the SARS infection, not COVID, SARS infection epidemic in 2003. Okay, there are room for many novel coronavirus. I know this class, Srivatsan probably is teaching you combinatorics and mathematics and statistics, how many combinations one can get by mixing and matching these genomes. Sky is the limit. So we can't be prepared. We can only be knowledgeable about these things. That's my take from this particular content of this slide. In the next slide, okay. So what do we know about the genome structure? I just talked about recombination. So what do we know about the genome structure? So in the top you see right here, this is the complete genome, and there are structures. This blue part, or blue-green part, if you might, that is the structural protein part. It doesn't change from virus to virus because that is critical for making the shell, the round shell that the virus has, inside which they have the RNA genome. Okay, the protein that make the round shell, that fairly conserved across coronavirus, at least in the viruses that we are dealing with, which is called beta coronavirus. Okay, there are 4 families of coronavirus: alpha, beta, gamma and delta. It's a beta coronavirus that we are talking about that causes upper respiratory distress in human being. Okay, and then in the top, in the genome, do you see the small part here, pink and then gray and red? Okay, This region is the most variable region across beta coronavirus. So what does they do? There is a S. I don't know whether you're able to see it, but there is a S domain, there is a E domain, M domain, and an N domain. We have to focus on this S domain because this S domain is the one that makes a protein that binds to human ACE2, angiotensin converting enzyme to how we get sick. So this is the protein from the virus that binds to us. So we are going to focus on that. So in the bottom, you already see that this is the only region that's magnified. So this pink, gray and red, that's magnified in the bottom here. Okay. So I am almost 40 minutes, half an hour into the lecture, so I have half an more hour. So I'm hoping I can conclude something. A lot of people are typing in a lot, but I will come back to answering those questions and comments. Okay. So in the middle, in the pink background, are the SARS-CoV-like and SARS-CoV genome, and this comparison is only for this small region here, okay, because that's all we are focusing on. Okay, so when we take this is the first sequence is from human early stage SARS-CoV and from civet. They look very identical, so there is only one genome structure here. The second, under the beta coronavirus, the second alignment is the mid or late stage. You already see things are changing. The virus is going through evolution inside human being. And the third one is directly from bat, which is kind of different, and it doesn't it doesn't cause infection in human. And the last fourth and the fourth one is the MERS-CoV, which caused Middle Eastern respiratory syndrome back in 2012. So you see that there is a lot of variation in this S domain. So what this tells you is this is what you want to focus. At least we know from the SARS epidemic back in 2003 that which region of the genome we need to be focusing. So that knowledge we can use now to understand the COVID-19 virus, which is called SARS-CoV-2. So from this work, we know where to focus on the genome and how we can quickly come up with regions for which you can do antibody, for which you can raise vaccine. So we need to do that very quickly because this pandemic is big. So this is something we need to learn. So I'm going to go to the next slide. And of course, people have done the crystal structure of SARS-CoV S domain with the receptor of human ACE2, and they know exactly how it binds. Okay, so the top in the crystal structure, and people who are not familiar, these are atomic detailed protein structures where the SARS protein is binding to our receptor protein in human. So the top green one is human ACE2 structure, folded structure of the protein, and the bottom cyan and violet are the protein structure of the S domain from SARS-CoV, which caused the SARS epidemic, not from the recent one. So we know that in which amino acid and which regions of the S3 is actually is an antigen that presents to ACE2. We know that from the crystal structure, which is a good thing because this helps us to map what may be happening in COVID-19. The next slide, I'm just zooming in the area where the interaction between ACE2 is happening with S1. What are all the amino acid that is interacting here with ACE2? ACE2 is not changing because that's a human ACE2. But what is changing is on the RNA, on the while RNA virus side, the amino acids can be changing to cause different kind of infection. For example, we are all familiar with flu. I'm going to deviate a little bit because we are all familiar with flu, because this can be heavy for people who are not familiar with the three-dimensional structure. I'm hoping my class is familiar, but people who are not, for them, in flu what happens? Every year you are vaccinated like other diseases, right? Why? Because the flu is an RNA virus, and they mutate. Every season they mutate, because every season it makes a lot of human beings sick, and every human being who gets sick with flu is a potential reservoir for these RNA viruses to mutate, because it replicates. That's why you get sick. There are too many million molecules, million, or even however many viral particles are made in your body. That many times it has opportunity to mutate, and because of that, by the end of the season, you have new novel mutations, and we know how to make a vaccine to figure out which one is going to be epidemic next year. So we make vaccine. Similarly, in coronaviruses, these amino acids keep changing, trying to find different ways to infect people. They are not changing because they want to infect people, but in the process of changing, we do get infected. That is called natural selection, correct? The virus is not planning to make it sick, okay? But they are mutating randomly inside the human body. So suddenly they may mutate in a way that makes us very sick. And when we get sick, that particular mutation gets selected because it's now starting to multiply. Okay, that's what natural selection is all about. So it is not, the viruses are not conspiring against us. Okay, so now we know which amino acid or which amino acid in COVID-19, COVID-2, SARS-CoV-2, we need to focus on to make antibody to treat that. That's what we are learning from the previous SARS epidemic. In the next slide, I'm showing. Now this is a kind of heavy slide, but I put that here. Hopefully I can make it simpler. Okay, on the right, do you see on the right there are amino acids? These are amino acids, and my class definitely is aware of what amino acid sequences are. I want to tell you, these are not protein sequences. Every column, so there is a matrix of characters here, right? These characters are single code amino acids, okay? And these characters are not a protein sequence, but each character is a position in a protein, okay? Each column is a position in the S protein that is changing across many species we have sequenced. So these 40 amino acids are not linear in a protein chain, okay? But these are the only places where it is evolving, changing, okay? So this is a good representation. Maybe I can go back to this again and explain. I hope you guys are getting it, okay? So this kind of reduced representation of which amino acids are changing and how they are changing is helpful in understanding how evolution happens inside a human body. I just told you a few minutes ago that every human body that gets sick is a reservoir of changes. And this is kind of humbling. This slide makes you humble as to, oh my God. If you didn't think about, oh my God, then you did not understand this slide. Okay, so try to understand this. On the left, I'm going to have to read it. I hope you are able to read it on your slide, because you guys must be sitting too close to your slide. Okay, so what is it showing? On the top three rows in the sequence alignment on your right are the civet and the raccoon dog from which we sequenced it. Okay, so they are very identical. and that was also very similar to the first few patients when you sequenced it. So in the top, then two raccoon dog and lot of civets from animal market with low pathogenicity. Okay, these are all low pathogenicity. Now we know, because we have the viruses, we can see whether they cause high pathogen or low pathogenicity in animals, and so we know this answer. Okay. So all the way you go to civet, and suddenly you see in the second column on the matrix, you see patient. Okay. Now this is a patient from Guangdong, December 2003. Remember I told you this epidemic started in 2002, 2003. So this is from a patient which is a low pathogenicity, and this is right here. Right next to it is a sequence. Already it has mutated, and already you see how the mutation from the top to bottom keep on mutating. So in the middle somewhere is the low pathogenicity mutation in human, and then as you go down in the bottom, it's all from patients at different times, from different people. This whole, the fourth column you see, you have the high pathogenicity mutation. Okay, and you already see so many positions got mutated, and in the end you have the epidemic when in all 40 positions. This is not a totally different protein. I'm going to hit back because this can be confusing. This is a long protein of 500 amino acid. What we are showing here in this alignment is only the 40 positions that are varying across all the S1 protein that we have aligned. So in all these 40 positions, it has changed before it become an epidemic. So all these mutations are happening right as we are getting more and more sick. So this is the crazy thing. So this slide takes home the message that yes, you start people start getting sick with a particular viral strain, and then of course it keeps mutating. So by the time the 1,000th patient got sick, the virus has already changed, and so it can go from low pathogenicity to high pathogenicity to epidemic to pandemic. So that is what is happening now. So I'm going to go to, I hope that you got it. If you didn't get it, I can go back in the next lecture. Okay. So now we know how much we know about these things, how they mutate. Every sick animal and sick body is a reservoir of natural selection. And natural selection is, naturally these are mutating, and selection is happening within the animal because a random mutation, it so happens it gives virus an advantage to make the person more sick, and if the person is more sick, they replicate more, and that gives it opportunity to even mutate more. Okay, so that's where natural selection is. Of course, one question you can ask is, why does the natural selection doesn't go to kill the virus? Because it's mutating so fast it can die. That may happen, but you won't see it. They're dead, right? That's a natural selection is a crazy thing to understand. You only see it when they are reproducing, when there are more of it, when you get sick. When the virus die, you don't know them because they're dead. You don't see them. You cannot identify them. You cannot sequence them. That's the thing. So it's a one-way street. Okay, that's why it seems like, why is it doing it? Because the other half you don't know. Okay. So from bats to animals to human, because we know now, just to summarize what we learned, bats directly from coronavirus from bat doesn't make humans sick because bats don't get sick from coronavirus. The answer to that question I still don't know, but I guess one way I can understand is there is a lot of microbiomes in our gut, okay, which can make us sick when it goes in our throat. But we don't get sick, but we are harboring it since our childhood. But if it goes again through our throat to the stomach, we'll get sick. So that's what is happening. Bat gut may be harboring a lot of coronavirus just for some reason, and they are not getting sick unless it goes through their throat and everything. So for some reason they are not getting sick. So we do need to find out. But when it comes to animals do get sick. That was my first question when I asked, if the animals are getting sick, why are we not finding those animals and why are we trading those animals? Can't we tell an animal is sick so we can avoid those sick animals from transporting it or getting it in our market? And that I thought is a crazy thing. So each animal, because the animals get sick, they become the reservoir now. From bat, either the animals eat bat or something, they get sick. But when the animals get sick, they now become a reservoir for new mutation. Of course, they are not mutating it to make humans sick, but humans are next to them in this market. So sometimes if a viral shedding or spillover is happening from an animal that you're handling and you're next to it, and unfortunately it got a mutation that can bind to your AH2 protein on your lung, you get sick. Okay, so one thing to know here is, now the question is, okay, there was a news in COVID-19 epidemic or pandemic at that time, I don't know. Yeah, people are getting sick, but there is no human to human transfer yet. Okay, what does it really mean? Means that for a long time, animal handlers may be falling sick and may be dying. This can happen for years. For example, in the case of MERS, what happened was for 30 years, camel handlers were getting sick, but their families were not getting sick because they were not passing from human to human at that time. But constantly for 30 years, humans who were handling camels, they were getting sick, or eating camels, they were getting sick, but sick person was not transferring from to their family. There was no human to human epidemic at that point. But now all these sick humans were now reservoir for natural selection in human. Okay, so suddenly a mutation can become really bad and starts binding to AH2 really strongly, and then this binding so strong that now it can go from human to human. Okay, so it did not happen suddenly. In nature, it did not happen suddenly. That's what I learned. In nature, it happened progressively, but we did not notice it because we are not studying sick animals, which I think we should be doing. Okay, I'm almost at the end of an hour, so let me see what more I can tell you. I'm going to the next slide now. So now we know a lot. We know where exactly in the genome when we sequence CoV-2, we know where exactly to focus, which amino acid to focus, although we don't know how it folds. But based on the homology to the S domain of SARS virus, we can tell which amino acids are important. So we can know a lot. And how did we find out it goes from bat to pangolin to human, correct? So in the next slide, of course, okay, sorry, you went too far. So when we sequenced, so let's take a step back. So what did we do? We know a lot about coronavirus and SARS. We sequenced the SARS-CoV-2 from a patient and then from human, the top, and we straight went to the S3, S1, S2 junction, the S protein I was telling you, okay, where most variability happens. So we know we don't want to waste time with the rest of the genome. You know, the genome is 30,000, 29,000 and odd long, but we know where exactly to focus from our previous effort. So we know that in the SARS-CoV-2, there is an insertion of 4 amino acid right here, which is not there in the bat version of the thing that we have identified. I'll tell you where we got that. We know that it's not in the pangolin, from which we know COVID-19 started, because the people who were handling first got sick where they were handling pangolins. That's how we got the pangolin genome. But even from the pangolin, the first human, when they got sick, they have this change in the 4 amino acid. Do you see that? The 4 amino acid change here, insertion. Okay, so that created O-linked glycan residue, which is heavy for the end of the lecture right now. I can explain this in the next lecture, about the significance of this in terms of COVID-19 pandemic scenario. In the next slide, we know that an insertion, a similar insertion what we see in COVID-2, enhances cell-cell fusion, in lab experiments. We know that efficient cleavage at the same site in MERS-CoV makes the bat COVID infect human. Okay, we know that that site is very, very critical. Okay, and we also know a similar cleavage in a different protein called hemagglutinin in avian influenza virus converts the low pathogenicity virus into a high pathogenicity virus in the same site, in a very homologous site. Okay, and we know that the O-linked glycosylation site that got created with this insertion that I showed in the previous slide, the 4 amino acid insertion, can give the virus a protective shield from human immune system, because these glycosylation sites hides the protein from human immune response. Okay, so these things we know. So we are very curious about the 4 amino acid insertion. So I think I am going to stop my lecture here, because I want to, the next few slides are about how rare this 4 insertion is. I want to emphasize on that. These are very rare insertion. The only way it can happen is by natural selection in an animal where it is doing nature is doing an experiment within a human body, and randomly which is mutating. This kind of selection can only happen. So next few slides are to emphasize on that point, which is very technical. So I am going to skip to one very interesting thing that I thought. I found it jarring that we are not taking care of sick animals. With this slide, I will stop the lecture. It will take me few more minutes. I started 5 minutes late, so I'm going to take 5 more minutes. So what is this slide showing? Okay, when I asked the question that, pangolins were sick in the market and people handled it, then people didn't know they were sick, or they didn't care they were sick. So where are the pangolins coming from? And how are they being transported? When you look at the pangolin situation, what makes me mad is pangolins are endangered animals, and the biggest trafficked animal in the world is pangolins from Malaya to China. The biggest trafficked animals. And because this is endangered, trafficking is illegal. So what was happening I'm going to go to my screen so that I can come back to see, because okay, so the pangolins are being traded, okay, and so in the customs in China, they're catching this illegal trade, and when they're catching it, what they saw is those pangolins were really sick. In the market, they're sick, but people are not caring about them. But in the customs, when they catch, those pangolins were dying. They were sick. They can tell. And they said, okay, what are they dying from? So what happened was they took large number of pangolins that were dying. They post-mortem took the lung swab from those pangolins and they saw how many viruses are there. Of course, this happened before the COVID-19. We didn't even know pangolins were going to eventually be traded and we are going to get COVID-19. Believe it or not, this publication that I read was in October 2019, only 1.5 months before this pandemic started. And this paper says clearly that coronavirus and there's another virus called Sendai. These are two families of viruses that were maximally present in the lungs of these pangolins that were dying. So, yeah, what can we do in future? Of course, you are not going to be able to imagine the sequence or the mutation that these coronaviruses can acquire to make us sick in future. But my thinking here is we need to change our behavior. We know that our human behavior is doing this. We can protect ourselves. We can care about the sick animals. We don't have to transport sick animals and get so desperate about selling them in the market. These pangolins were sick even before they got to the market. So the coronaviruses is one of the reasons why they could be getting sick. We don't know more about it, but we do know our behavior. This was 1.5 months ago. This came in a publication, and this publication warns people that pangolins can be carriers of coronaviruses for future epidemic or pandemic. And within 1.5 months, we saw COVID-19. With this, I'm going to end. I know all of us probably read a lot, and I'm going to go back now to quickly see. There are a lot of comments, a lot of questions, and you guys also see it on your chat box, what others are asking. So let's go back, start from the beginning, first come, first serve. Okay, so one of Nisha one maybe. Ma'am, I think the connection with bat we know because in China they consume bats, so they monitor them and find that some symptoms were caused by this virus. Okay, that's good. Thank you. Okay, I didn't know that they eat bat. I didn't know that the markets even hold bat. Okay, and why is bat very common for most of the virus transmission? That was Karen, and because bats are the hotbed. Okay, that's what we've searched. Since 2002, we have 18 years to look into coronavirus. We looked into many animals. I am not very familiar with the entire, I'm not a virologist or microbiologist, so I don't know the extent of research that has happened, but it's open. Lot of research has happened, so one could find out why it's only bat. Then, is it a general tendency for viruses to have such hosts which are not affected? Perhaps. How would we know if we are not affected? We are not looking for them. See, in bioinformatics, I always say, for human activity, there is always a cause. Unless it makes us sick or it makes us rich, we are not going to look for them. So yeah, it is possible. There is no incentive for us to look if we are not affected, correct? Okay. What if, having said that, I will tell you there are some virologists, there are some drug companies who work on viruses. Throughout these 18 years, they have been working. Okay, when people didn't understand why the hell they're working on the viruses. Now they are suddenly on the spotlight and they like what they're doing because it actually connects their drugs and everything to actual COVID, right? So then Nishchit or someone, what if we infected patients under a lot of stress, will the viruses come out of the body and individual get cured? Shedding doesn't mean that you're shedding all of it. Okay, yeah, that will be great, right? That will be great if you can shed all of it. I think stressed people get infected very quickly. That explains why older people get sick, because their body is already stressed by diseases, other diseases, and lower immune system. So, and then Pushpinder. Hi, Pushpinder. So, hope you're still there. So Pushpinder is answering that question now. Report says that there are multiple reasons why bad carriers so many viruses, that includes food choices, colonial or solidarity nature, popular. So you guys are reading it, so I don't have to read it. Pushpinder has the answer here. And then you have hello ma'am that just joined, I guess. And then how virus mutate, what types and stress signal lead to mutation in human body. And okay, this is easy, because I told you one reason you get sick is because your immune system did not detect the virus. The moment the virus, and also I told you the glycan site that hides the virus from our detection by our immune system, so you don't get sick. So they get in. So when they get in, by evading our immune system, they start multiplying, they make more of it. And RNA viruses are 1,000,000 times, mutation rate in RNA viruses is 1,000,000 times more than in human, more than in any other host. Okay, so replication, every replication is a potential opportunity for mutation. So if something replicates 1,000,000 times, obviously it mutates. And many of them die, because mutation also can be bad for the virus. They can die, but it also gives an opportunity inside a host for selection. That's why inside a host only these selections can happen. Okay, so it's hard to predict in the lab and everything. Okay, so but what makes the beta coronavirus to mutate at such a very, all viruses mutate rapidly. I told you about flu viruses, why you have to take vaccine every year. They all, RNA viruses are crazy mutation. They go through crazy mutation. Every year in every year in case of influenza, we generally take some medicine. Yeah, medicines. The medicines are not working in COVID because they are mutated too much, and there are some medicine which work by mechanism of how it kills the virus. For example, retroviral, HIV. We are using HIV. The 2 drugs in HIV, they are in clinical trial. You go to NCBI site called NCBI virus. Okay, that site gives you a link for clinical trial. There are 128 clinical trial going on with existing drug in the use of COVID, and doctors are already using malaria drug, HIV drugs. HIV drugs inhibit the retro translation of RNA to DNA. There are HIV viruses that inhibit the protease from cleaving, which is required for viral particle to assemble. And malaria drugs changes the pH of the cells. When the pH of the cells change, then the viral particles are not able to assemble. Okay, so all these things are inhibiting at different stages. So there are 124 clinical trials happening, and doctors are treating patients with this drug when they're really sick, but nothing has been approved. So I think beginning of May we are going to see some clinical trial results, and at that point I think we can all be happily taking some drugs. Okay, so I'm going to stop here. You have all these questions, so thank you, and thank you so much. Thank you, Anna, yeah, for staying all through this. So that's my sister, is the last one great lecture she is saying. That's my sister from Houston who joined us. She's a doctor, medical doctor, and we have been talking a lot. So thanks guys, bye now. We'll talk about it again.